Yeah, good morning. So today I'm going to say about the vaccine. So what is member vaccine? So here, vaccine can be say that it's a type of immune system. It will boost our immune system. It will stimulate our immune system. So here, in olden days we don't have a vaccines. So now we are called as vaccine is nothing but a live attenuated killed suspensions, live attenuated or killed suspensions of microorganisms. So here basically these microorganisms we have a two types we can use it. One is bacteria and virus. So based on that bacteria, it's called as we can call as bacterial vaccines. So we are using as a virus as a vaccine, it's called as viral vaccines. So first to invented vaccine is rabies vaccine. So this rabies vaccine is for the treatment of rabies disease. They invented for the treatment of rabies disease. So here I'm going to say that what is member vaccine here. So basically when you're going to say about the bacteria is nothing but a microorganism, virus is nothing but a microorganism. So these virus will cause the diseases to the human beings. So bacteria also will cause the disease to the human beings. So here this vaccine can be prepared. So actually when you take the virus or bacteria, so they have a particular cycle made up of the component biomolecules, I can say that. So these biomolecules, so what is say that biomolecules are nothing but proteins, carbohydrates, nucleic acids and lipids. So these are the biomolecules I can call as. So these biomolecules, any organism is made up of the biomolecules. It will be composed of the proteins, nucleic acids and proteins and lipids we can call as. So our cell organelles, everything is made up of the with the biomolecules. So here this biomolecule, I can say that these biomolecules it won't give the biocompatibility to the other organism. It won't give the biocompatibility to the other organism. So we took the here vaccine. So first we'll go for the what is that we have a types of vaccines we have it. So here we have two factors we will have it. So this is called as pathogen. Suppose when your bacteria or maybe virus will be entering into our body, it will stimulate our immune system. It will stimulate our immune system. Our immune system has been responded. Otherwise, it will cause disease. In this case, you are taking a particular virus. So your virus or bacteria will have two characters will show that. One is antigenicity, other one is pathogenicity. So antigenicity means it will stimulate our immune system. It will recognize our body will recognize that the host is came. Our body will recognize that the pathogen came since our body. It will recognize as foreign molecules. It will recognize as the foreign molecule. These after getting into our foreign molecules, I have a different types of immune system. We have primary immune response, secondary immune response. First, our primary immune response will act against those molecules. So primary immune molecules we have it, like our thymus and lymph nodes. They'll act on T4 cells, T killer cells, T helper cells. Those cells will activate and they'll fight against the particular bacterium or virus. So here, this bacterium or virus, so whole organism. So suppose bacteria have it. So bacteria will made up of bacterial membrane, and we have inside we have a nucleic acid. We have it. Nucleic acid means genetic material. We have it. So both the components will not be act as a pathogen. So some of the components will act as a antigenicity. So what you're doing here means you are going to remove your bacteria, maybe virus. Suppose best example I can say that. Suppose virus we have it. So virus is made up of capsid, and inside we have a nucleic acid. So then it will tail and tail fibers we have it. So these sometimes your capsid will act as a pathogen. Otherwise, sometimes your DNA will act as a nucleic acid will act as a pathogen. So in this case, you are going to remove the pathogenicity. So here, when you are going to remove the pathogenicity, remaining part we can use as a vaccine. This is called as vaccine. So here, it won't show the pathogenicity. Otherwise, those we can save in our body as a memory. We can say that. So here, I am going to say that. So your your virus, maybe bacteria, maybe live attenuated. Means you're going to attenuation. You're going to fix your bacteria or virus in attenuation condition. It won't cause the pathogenicity. Then killed suspensions. So here, before going to say about the killed suspensions, so we have it. Suppose here I said genetic material. What do you have it? Then capsid we have it. The protein capsid we have it, and genetic material we have it. So we can remove the genetic material. Sometimes your capsid may act as a pathogenicity. Suppose you remove the protein coat, the DNA will act as a pathogenicity. So here, so you're going to remove the particular pathogenicity of the viruses, then you can use that one as a vaccine. So these vaccines can be made up of different types we have it. So one is live attenuated vaccines. Live attenuated means your virus can be attenuated. Attenuation means you're going to put in an attenuation condition. Simply, best example, rotavirus, or MMR, nasal flu vaccine, and chickenpox vaccine. This is called as live attenuated vaccines. So here, so we can call as virulent factor, non-virulent factor. So before going to say about the live attenuated some of the vaccines, killed vaccines, and different types of vaccines I have, I can say that's one. Types of vaccines I can say that. So here, virulent factor, non-virulent factor. Virulent factor means it will cause the disease. Non-virulent factor it will stimulate immune system. So simply we are going to use both the molecules. So what is the you needed? What is the desired molecule you want to use that? So then those molecules you are going to use as a vaccine. So the live attenuation means you are going to remove the pathogenic factor. So virulent factor you are going to remove that. It may be protein, it may be nucleic acid. So basically, so your protein may act as a, sometimes it may be your protein may act as a pathogen, or maybe nucleic acid may act as a pathogen. So here, before going to say about, so any foreign molecules, your best example, I can take that. Suppose transplantation, we can say that. You can say the organ transplantation. So before going to transplantation, we're going to check the genetical compatibility. You're going to check the compatibility of genetically compatibility of particular human beings. Otherwise, we don't transfer that organ. So when you want to, so a patient had a problem with the kidney problem, he suffered with kidney problem. We don't transplant. We are going to check the first. We are going to check the genetical compatibility. Then after checking genetical compatibility, we are going to transfer the particular kidney. It will be fixed or not. And so if the compatibility is there, genetically compatible is there, we can transfer the one patient to other patient. This is based on that one. So otherwise, we don't check the genetical compatibility. That will ultimately give the grafting. Because grafting means here, so you use immune system. So immune system will recognize this molecule, or the organ you are transplanting, it will recognize as a foreign molecule. It gives the production of antibodies. It will give the production of antibodies. So these antibodies will act on the particular molecule. Ultimately, it will cause the severe side effects to our particular human being. Sometimes it may lead to the death of the particular people. So this is called. So here, why I am saying this means any foreign molecule, it may be living organism like our bacteria, a virus. It will have a protein coat. It will have a nucleic acid. Sometimes it will cell membrane will have it. Bacterial plasma membrane we have it. So those molecules will stimulate our system. So what will do here? So when you enter into your body, so when a virus or pathogen will enter into our body, it simply to insert this DNA. Simply to insert this DNA to our cells. It will contaminate our DNA. It will contaminate our DNA. So ultimately, our DNA will contaminated DNA will replicate instead of producing our own molecular DNA, it will produce the host DNA. So after then it will produce host DNA, it will go for the protein synthesis. Ultimately, we will get a new molecule. So the new viral molecules, some bacterial molecules produces. Ultimately, patient will get an infection. So here, in this case, so vaccine, we are taking your DNA, you are taking the DNA of the particular bacteria, or maybe you are taking the particular DNA protein of the particular bacteria, we can use as a vaccine. That's called as. So here we are taking one organism. Those organism can be used as a live condition. It can be used in live condition. So you are going to put it in attenuation condition. We are going to remove the pathogenicity. We have removed already pathogenicity has been removed. It may be protein or DNA. Okay. So these pathogen molecule we have removed that from the bacterium or virus, that is called as live attenuated. Next one is inactivated vaccines. Called as inactivated vaccines. First inactivated vaccine is rabies vaccine. Then next one is botulinum pertussis vaccine, flu vaccine, and hepatitis A vaccine. They are called as the inactivated vaccine. So here, you are going to inactivate the particular pathogenic factor of your bacterium or virus. So you are going to treat that one. You are going to treat with your bacteria with a heating condition, not radiation. With radiation we can treat it. Otherwise you can treat it with formaldehyde. We're going to use a formaldehyde. Formally we can use it to remove the pathogenic factor. So we're going to inactivate that particular factor. So but it is in the other protein moiety and other nucleic acid moiety will be active condition. So here both the molecules are it will cause the disease, means you are going to partially demoralize that one, you are going to partially deactivate that one, then you can use it as a vaccine. That's called as what is that? Inactivated vaccine. Next one is subunit vaccines. Subunit vaccine means we are going to use the adjuvants. Suppose some cases the protein factor. I took the protein factor. This protein will not, it won't be effective. It won't effective to act as a vaccine. So in this case, we're going to add adjuvant. Adjuvant is supporting molecule to make a complete antigen. It's called as, it will stimulate our immune system. So ultimately it will act as a vaccine. This called as subunit, means you're going to here protein we can take it here, above nucleic acid we can take it here, other lipid moiety we can take it here. So other moieties rather than the one protein moiety, so you're going to add the carbohydrate moiety also we can use that. So this is called as subunit vaccines. So these subunits means, so here you can use the adjuvants. The adjuvant means incomplete antigen. In case of immunology we can say that adjuvant means incomplete antigen. So this incomplete antigen, you are making the complete antigen by combining the protein, otherwise by combining the nucleic acid. That's called as adjuvant. Here, so two subunits. Here this is an adjuvant, this is an protein or maybe DNA. We are going to combine it, we can use that as a subunit. This is called as subunit vaccine. Then next one is toxoid vaccines. So simply I can say that live attenuated vaccines, first one is live attenuated vaccines, inactivated vaccines, subunit vaccines, toxoid vaccines, conjugate vaccines, viral vector vaccines, and nucleic acid vaccines. These are the types of vaccines. So now I am going to go for the toxoid vaccines. Toxoid vaccines. Suppose some of the bacteria, they enter into our body, so simply they will release toxins. Those toxins may cause the severe disease to our body. Best example, tetanus, Clostridium tetanus. It will cause food toxin. Sometimes it may cause the death of the people also. So here we can use what is that? Toxoid vaccine, diphtheria and pertussis vaccine we can use it. So pertussis vaccine we can use it. Here recombinant protein vaccines we can have it. Recombinant protein. This is called as what is that? Conjugate vaccine. So you are going to make a conjugation. So one virus, other virus. So one viral factor, other viral factor. One bacterial factor, other viral bacterial factor. It may be protein, it may be DNA. So you're going to conjugate with a different molecule, different pathogens. We're going to conjugate, make it as a vaccine. That's called as conjugated vaccine. Next one is for DNA vaccine. Here DNA vaccine means nucleic acid vaccine. So here we have different types of vaccines. We have it. So we can use mRNA. Best example, mRNA vaccine is COVID-19. This is made up of mRNA vaccines. Otherwise, what is our single-stranded DNA vaccines and double-stranded DNA vaccines. So best example we can write a COVID vaccine. Particularly we can write the COVID vaccine as a mRNA vaccine. Next one is viral vector vaccines. Viral vector vaccines. So the viral vector vaccines means here I can say that replicating virus, non-replicating. So you are going to take the viral vector molecules you are going to take. Vector means transferring molecules. They will transfer the particular factor we are going to transfer. So we are going to vector, we are going to take it here. So these vector we are going to insert your what is that? Vaccine molecule. Vaccine means protein or maybe DNA. So we are putting inside the protein or DNA in this one. So we can use these viral vectors as a viral vector vaccines. Then next one is we have edible vaccines. So we can, new one is edible vaccines. Edible means like our banana. So we are going to insert the particular gene of our particular protein factor. Otherwise you can insert our DNA factor. Those called as edible vaccines. So these are the types of vaccines we can use it. So these vaccines will stimulate our immune system they'll make immunomemory cells. It will make that immunomemory cells. Suppose, best example I can treat with smallpox. So in a child condition, when we are in childhood days, sometimes we have infected with smallpox, but it can be treated cured by 21 days. We don't take a vaccine, anything, but those viruses will be cycling in our body. Our body will recognize that, it will produce antibodies, finally make that immunomemory cells will be placed in our body. So this is the use of vaccines. So the vaccine can be used different types of diseases. So COVID, HBVs, HIV vaccines. We don't have HIV, HBV, HBV, HBA, B, then Borderline vaccine, smallpox vaccine, polio virus vaccines. These are the means we can use the molecule. What is the molecule? It's the living molecule of a bacteria, maybe virus. So both we can use it. We can remove the only pathogens. So simply we can call as. So can you define that vaccine mean live attenuated or killed suspensions of the microorganisms? It's called as vaccine. So with these vaccine can be used for our human to protect from the prevention is better than cure. So simply we can say that prevention is better than cure. So that's why simply we are going taking the vaccine to cure the particular entry of the to stop the a particular viral molecule, some bacterial molecule entry. So whenever it will be sorry, it will be entering into the body. So ultimately it will cause the disease. Suppose we took the vaccine. So these molecules we have it. Those molecules recognize the particular bacterium or virus. They'll phagocyte that one. They'll fight against that molecule. We can get the immune system. So your immune system will be given with particular virus or maybe bacteria. So these are the use of our vaccines. So now we are using the recombinant vaccines we can using. So now technology is improving. So science and technology is improving. So we are using the recombinant vaccines. Recombinant vaccine means you are going to include 2 or 3 subunits. We are going to include the 2 or 3 subunits of the different viral factors. You can use as a vaccine. This is about the vaccine.